sorry.
Yeah, you should own that.
I'm sorry at all. And by the way, Rachel, I don't remember anything we talked about on Friday, so you're going to have to recap. Okay, so I'm better today. So I wanted to, this is a little bit of we're really excited actually to have Dr. Elizabeth Neumann with us today, who is assistant professor at UC Davis. And this is a little bit of an ad hoc CIDMI, or Center for Infectious Disease and Imaging seminar, along with experimental medicine. And Dr. Neumann is, we had this a few weeks ago through Amanda, actually. There's Amanda. Sorry. Who is working a lot with on the digital spatial omics side, but also looking at novel ways of looking at brain and heart in people with HIV, substance use, things like that. And I actually made acquaintance with Dr. Neumann, and I think there's actually a lot of shared expertise and interest with experimental medicine, and try to introduce the folks that will be interested as well. So bear with us afterwards as well.
Oh yeah.
So Dr. Neumann, after undergrad in chemistry at Baylor, went up 2 years at Urbana-Champaign to do her NSF, a Sophia for doctoral fellow, did her PhD work, and then I did a postdoc at Vanderbilt before landing here at UC Davis, and they're very lucky to have her. And today, Elizabeth is going to talk about spatial multi-omics towards understanding human health and disease. And with that, this might be a little bit new for a lot of us, and we're going to break out a little bit outside the ID sphere as well, but I think there's a huge amount of potential collaboration, also crossover with what we do as well. So this will be very exciting. If you have a question, please feel free to ask during the talk. I will pass the microphone around so you don't have to read it. Questions for people. Cool. Without further ado.
Thank you. So, I think this is on. Yeah. If you all can't hear me, let me know. But I teach massive undergrad classes, so I usually am pretty good about screaming very loud. So, two things before I get started. One, I've been there. I've sat through the worst talks of my entire life going, I understood slide 1, and then got to slide 2, and suddenly they switched languages, and I don't understand anything. And so I give people, especially trainees, the opportunity to answer questions or to ask questions during the talk. So remember, I'm a faculty member. I'm regularly on qualified exams and thesis defenses. So this is your opportunity to get back at a faculty member. To ask them the meanest questions you can. Or if you're confused, I'm happy to talk through anything. I'm not in love with my slides. We go quickly at the end. I keep track of the clock. You have your food. I won't keep you forever though. So any questions, just feel free to ask me. We can stop and discuss. No question is dumb. No question is bad. Right? And then, I remember number 2. So we'll get started. So here are some of my favorite images. They're my students' first images. And so when we think about spatial methods, often we hear talks from people that go, yes, they do the brain or they do the kidney. That must be the only thing they can do. So I want to encourage you to actually think about what you can use or what you can image. You can really span most things. In fact, I'll show you things that are itty bitty, like extracellular vesicles. And I'll show you things that are entire animals. Also image electrodes before. At some point, if there's a compelling reason, you make it work. And so these are some of my students' first images. So I want to encourage you that this technology is something that anyone can learn and anyone can use. So think about how you can incorporate some of these spatial components into your data. So with that, I'm a chemist, but I really like clinical science. So I always ask, how do I understand biology or clinical applications? And so as a chemist, I think about it from biomolecules or biomolecular classes. And so I put this central dogma of biology where DNA is transcribed to RNA. RNA is translated to proteins. And this is great, and I understand for the more clinically minded that these pathways are very confusing and more like this. But again, I'm an analytical chemist. I just build instrumentation. So we're going to go back to my cute drawing, actually. I'm more comfortable with this. But since I am talking to you all today, I've tacked on metabolites here at the end. And most of what I'll be talking about is actually lipids. Because these are things that we often forget about, but are actually interact with every single one of these spaces. And so because I have the room, you're going to deal with my new central dogma of biology. And so my day job is actually to teach large classes of undergrads. And so, like maybe some of you in the room, they always hear my brilliant lectures and then ask me what's on the exam. And so I'll give you the exam answers for the end of my lecture. So because we're doing Q&A during it, you'll get a Scantron to fill out on your way out. And so as an analytical chemist, I think about how do we study these molecular classes. So we go to DNA. You really have PCR and standard sequencing if you're old school or you're dealing with something that's very difficult, or next generation sequencing. And there's different types of next generation sequencing, but the backbone of these are actually very similar. It gets slightly more complicated when we look at RNA, specifically mRNA. You have all of these different renditions of RNA-seq, some of which are spatial, some of which are not. Some have higher dynamic ranges, and so on and so forth. But the underlying technology is very similar. You learn about the same parts of RNA. And then we go to proteins, and things start getting really, really dynamic. So we think about how do we study proteins? You have things like LC-MS that tells you the mass or the identity of a protein. Sometimes it will tell you their post-translational modifications. You got Western blot, which tells you its rough molecular mass, but then you can start looking at other things, and maybe you can figure it out in a complex situation. And then we have something very different like X-ray crystallography, which tells you its 3D shape. And so all of these things tell you something different about a protein. And then we go to the metabolites that, again, is very different, and I've tacked on to my cute drawing. And this list is massive. Every time you try to look at metabolites, you use a different instrument. And it tells you a different component of the metabolome. It covers different classes of the metabolome or different function, whether it's a mass or what they're doing, or maybe what their functional chemical components are. But my point is, this list is not even comprehensive. So as we move down the central dogma of biology, our chemical information gets massive. So then I stay up at night. Even last night, I found myself awake at like 5 in the morning asking, how do I study biology then? Again, this list is not even a real list. It's not comprehensive. And so again, how do we measure it all? This is actually the crux of my lab. Is can we mix a lot of these different technologies together, and actually can we do it all in one instrument? So I'll let you think about that last part of it. So I'm at University of California, Davis. Here's a token picture of myself in my office. I believe a lot in the ability of communicating science with people who aren't scientists. So my parents are farmers. And so when I got this job, I called them. I'm like, hey, Dad, I'm going to go work at UC Davis. He goes, oh, you're going to do some more science? I go, yeah, I'm going to do some science. I'm an assistant professor. He goes, you've been doing science for like over a decade. And I go, correct. You are onto something there, sir. So then I like got my office, and I was like, my office has a door. My office is now full of toys. And so all of a sudden, this picture allowed me to actually communicate what this job meant to my parents. And so I do a lot of imaging, but thinking about how you communicate your science to a diverse audience is important. So if you're like me and your parents are farmers or someone that maybe isn't as attuned with science, think about, this one actually works quite well, think about how you translate what you're doing to a general audience. And then I actually am on the 5th floor of this building, which is a joke. So if you look for mass spectrometry course, they're usually on the ground floor or the basement, right? So someone goes, why don't we put the mass spectrometrist on the 5th floor with her instrument that doesn't actually fit in the elevator? So this is Bruce, our timsTOF Flex. I developed him as a postdoc and then got commercialized. And then, so I have like serial number 2 in my lab. He does not fit in modern elevators, even freight elevators. So my first experiment actually as an assistant professor was like, how do we do this? Because my space is on the 5th floor, because again, someone thought this was funny. So we called an elevator tech, dismantled the elevator, got him up on the 5th floor. We then decorated him because he's a shark. He's named after Jaws or Finding Nemo, depending on the demographic. It turns out the parents are just super afraid of the ocean, and so we have to be like, don't worry, don't worry. This is Bruce from Finding Nemo. Fish are friends, not food. Their kids, though, love the Jaws reference. They're like, oh, yeah, cool, eats this shit. And so I inherited a lab from a geochemist group. So I actually had to do a lot of renovations myself. Don't tell our facility. But if you're starting a new lab, for any trainees that are interested in this path, often you have to do things you weren't expecting. And renovations is often what delays people the most. So don't feel empowered to take a hammer and start knocking out stuff and making them for fridges. Again, you can dismantle whole elevators if you have the right personnel involved. So this is my lab. And more importantly, here's the people in my lab. So we started about 3.5 years ago. And I really just want to give all of the credit to the students, right? Keep in mind, 3 years ago, I didn't think I was going to get any students. Because I show up and I go, I got hopes and dreams and no money. And then somehow 3 students joined and they said, hopes and dreams. I think they missed the no money part. And they're like, hopes and dreams, hopes and dreams. And they keep getting more students. And I'm like, now the NIH is following my no money, but lots of hopes and dreams. So anyway, we work incredibly hard with very little resources. And we've done a lot of clinical science. So my students, upon their first year in the lab, get a Neumann lab t-shirt. It's a laser. The laser glows in the dark. And for every student I have, they work with a clinician. Again, this goes back to the communication of science. It's incredibly important to be able to do really cool chemistry, but again, in context. Yeah. Yes, they do. I'll make you one. You can be an official Neumann lab person. So they glow in the dark, and on the back is actually a giant shark with the year the student joined. So, Neumann lab sharks. So clinicians biologists can actually earn a shirt. These clinicians are partnered with my students, again, so we can do chemistry in context. And then how I move them in is I'm like, you can generate cool images. Also, you can get your own shirt after our first paper's accepted. It used to be after our first grant's accepted, but now we've pivoted a little bit. So now after we get a paper, the clinicians get a shirt. But joking aside, it's often hard to sit at the interface of the field because the language is completely different. But it's super important to train students to actually cross these boundaries so we can do some of this interdisciplinary science. So every student has a clinician. Their faces and names will be featured in the top right-hand corner of those slides. So again, I'll give a survey of what my lab is doing. If you have additional questions, we can talk about it. But if you see them running around at conferences and stuff, make sure to give them a thumbs up. Again, they're operating on hopes and dreams. And so keep them alive with the positivity and high fives. And so we use mass spectrometry. And specifically, we use matrix-assisted laser desorption ionization mass spectrometry, also known as MALDI-MSI, because my talk will be twice as long as I signal. So the gist is we take a tissue, we're given an electrode, we're given a whole animal, we then spray it with a small organic matrix. This serves as like an explosive. So when we hit it with the UV laser, we then explode out all of the endogenous molecules and throw them into the gas phase, and then charge them so they can be detected by mass spectrometry. And we're using a laser system that is actually tunable, so we can go down to cellular images. So these spots are about 10 microns wide, 5 to 8. You then raster this UV laser across our tissue, collecting mass spectra at every single point. For every peak, the discrete molecule we're able to detect. And we've actually maintained where on the tissue this mass spectra comes from, allowing us to build an image. So generally speaking, our workflow is we take a tissue, often it's fresh frozen, sometimes embedded, sometimes not. Usually we work with whatever has been collected at the time, so we're pretty flexible there. We then section and house to 10 microns thick. That's about a third of your piece of one hair, so it's very, very thin. Section at 20 degrees. We then take some histological images, because again, we're trying to add context to the data. So we take histology so we can get some landmarks and start embedding the data we're looking at. We then spray with our small organic matrix. You can think of this as a 3D printer, but instead of plastic, we're actually spraying chemicals. Again, we're going to then ablate it with fluids. So the benefit of mass spectrometry is we're high complexity. We're looking at tens to thousands and even tens of thousands of molecules per pixel. We're sensitive, we're at attomolar and femtomolar detections. That's, to put into reference, the best is usually even single molecule. Generally speaking, in our system, we're looking at 30 versions of the molecule. So, so we can look at very, very low abundance molecules to things that are in 10 billion. Our spatial, slides aside, they don't want to cooperate. So our spatial resolutions are between 5 to 10 microns, but I'll teach you how to cheat a little bit so we can go data there. And we can measure things that are hard to label, like lipids or glycans or neurotransmitters, glycogen, things that there's no antibodies for, they're not natively fluorescent. And so what I'll do now is I'll walk you through an example of what our data looks like so you can feel what the data is. So then when I go through the examples, you can kind of follow, we can spend more time on the actual cool applications. So here's the average mass spectra of an entire human kidney. Each one of these peaks is again a molecule we've detected. I put a label on it. We're primarily going to look at lipids today, not because we're limited to that, but because lipids are cool and no one has cared about them until we're interested. So on the far end, you have a PC, which is phosphatidylcholine. That's the head group that often controls things like fluidity. And then you have some numbers at the end, and that's the number of carbons in the acyl chain, as well as degrees of unsaturation. That will tell you curvature that the membrane is going to take, as well as some things about nutrition, which I'll talk about later. I've labeled some of these things. This is positive ion modes. So we're looking at phosphatidylcholine, sphingomyelins. There's some PEs here, but you can tell that the density of this data is actually quite big. I'm only labeling some of the more abundant things, but there's lots of stuff. This is actually only about a quarter of our data set. Now, the mass spectrometrist in me goes, look at how cool the data is. But again, we're trying to add context here. So we can convert this data to an image. Yep. The numerical meaning of the x-axis is the mass to charge ratio. So in our case with MALDI, we have a single charge. So it's actually usually just the mass of the molecule. And so the masses that always tell you what the molecule is, it's actually molecular. But we can do tandem MS to get structure. So I've added that. One that they have the question is, do we have a reference? Can a single line be more than one thing? The answer is absolutely. There's things like isomers, they are under the same molecule. Now we have a gas-phase separation that can separate isomers, which will come against various things. So they have to have different masses. In the case of lipids and small molecules, they often do. So here is actually the same exact data. This is a whole human cross-section of the human. So for the renal pathologists in the room or those that are listening, on the top is the cortex, the bottom is the medulla, and those yellow spheres are glomeruli. So glomeruli are involved in filtration of the blood, and then it gets passage to different tubular segments. Depending what segments it in, you get different absorption of nutrients. And so here's only 3 of the lipids that were featured in the other slide. So pixels in red have a high abundance of PA. Pixels in green have a high abundance of PE. Pixels in blue have a high abundance of PI. So as you can appreciate, there's very few pixels that are actually red, green, or blue, right? Life is a mixture of many of these different molecules. And even by visualizing 3 things, you can actually make it actually resolve vasculature, which is in yellow, or glomeruli, which is proximal tubules, orange, which is distal tubules, so on and so forth. So if you take a piece of tissue, homogenize it, you're losing all of this rich context that you can actually be getting. Again, we can play this game with other lipids here. So here's 9 of the 2,000 lipids we detected, and you get unique chemistry here. But as a scientist, you don't take my word for it. Here's thumbnails for about 1/10 of the data, where you have a mass in the top left, which correlates to a lipid, and then it spans from dark blue to yellow, or blue, the low abundance, yellow is high. So if you compare all of these thumbnails again with the tissue, you see different distributions of all of this chemical information. And again, a lot of it actually correlates to functional units, like glomeruli or medulla, or segments of these things. And so there's some biological function there that is completely unknown. So that's what the data looks like. That's how we go from line spectra to images. It tells you about some of the depth of our data. What I'll do now is talk to you about what we do with it, because that's actually the cool part. Oh, yeah?
Sorry, just making it. Is each pixel like one place where the laser lasted?
Correct.
Okay, so you get this picture for like each of those ones.
Correct. And then this would be example of the heat map for one of the so you can visualize it. So one of the things we started out doing is actually wanted to see if there was impact on diet on our income. So when I was a grad student, we actually couldn't really do nutrition-based studies. Because if anyone's in this space, you know that like the biological heterogeneity is massive. And our instrument heterogeneity was also massive. So what we basically did is throw our data up and go, yeah, this is really hard. So one of the benefits of the instrument we developed is it's actually very, very stable. We actually have reduced a lot of our chemical variability. So we wanted to give the nutrition diet-based studies a chance. So what we did is if we look at the differences between a control diet, think about what a mouse should be versus a Western diet, burgers and milkshakes, as well as sexes and axes. And if you actually just consider the whole brain without any subsetting, you get very little in the lipid space that's actually statistically different. It turns out this massively changes depending on where you are in the brain. So when we start subsetting our data into things like cortex, corpus callosum, or hippocampus, we start seeing some cool shifts. But I got stuck. So for those that understand neuroscience or familiar, I was very confused when we found nothing statistically different in the hippocampus. Because I'm going, that's the most plastic region of the brain. I would expect it to actually have dysregulated metabolism and actually be quick. So then I was actually a couple weeks later on a trip, you know, in the airport eating McDonald's, as I do. Because you have to reward yourself for getting up at 3 in the morning. So I'm eating my McDonald's and I go, God, this would be terrible if I ate McDonald's and then my hippocampus massively had made major molecular changes, right? You would suddenly forget a lot of things. So then I thought about it and I'm like, duh, you actually don't want your hippocampus to change that much with slight changes to diet. That makes obvious sense. Because again, I've actually been able to make all of my flights, and I think that would not be true. You have massive lipid change. So then I was like, God, there must be a cell that's buffer. And it turns out it's actually microglia and astrocytes. And I'll talk a little bit more about this in the context of disease. So we wanted to then extend this to Alzheimer's disease because clinicians have always seen that individuals with Alzheimer's disease have reduced cognitive decline when they're undergoing ketosis. And part of this is because they thought patients would forget to eat. Now, actually, I'm going to try to convince you that that's not true. It's actually biological, that you or individuals with Alzheimer's disease actually try to undergo ketosis on purpose. So here on the left is a mouse model of Alzheimer's disease. And this is the distribution of PI38.4, where blue or purple is low abundance, yellow is high abundance. If you do LCMS, this actually isn't a hit number. It's not distinct between Alzheimer's and wild type. Because again, the spatial context is important. So again, I'm not a neuroscientist, I'm a chemist. So I look at this and go, yeah, there's these speckle-like guys that's got to be A beta plaques, right? Like it's an Alzheimer's plaque. So I run like correlation coefficients, and it turns out it's like 87% correlated. And I'm like, that's not actually that high. Like it's high, but it's not what it should be. So then we did MXIF and we saved for a bunch of different cell types and different cell states. And it turns out that it's 99.8% correlated with activated microglia. And I was like, now we're on to something. So activated microglia are the immune cells of the brain. And so this lipid is highly correlated with that. So we're like, cool. We're spatially resolved. I wonder what happens if we actually look at the A beta plaque and look at multiple cell layers outwards. So how to read this is A is an A beta plaque that we've stained orthogonally, so we this is the plaque. A is the cells, and in this case specifically microglia within the A beta. B is one cell layer away. C is 2 cell layers. E is 3 cell layers, right? So we're getting farther away from this plaque. Here on the right, I've shown one plaque here. This is one, for example, because we do this for like hundreds of thousands. It turns out that's a really terrible visualization. So this is one, but the data is actually, we're doing it for all plaques 30 inches. So on the top left, the average mass spectra for microglia at that plaque interface. And again, there's lots and lots of cells represented here. And it turns out as you get farther, or again, in the reverse, as you get closer to the plaque, you get a high abundance of this PI38.4. And as you get farther away, it's actually exchanged by this SX ceramide. So I spent, as a chemist, a stupid amount of time trying to figure out how you convert these two lipids together. Because as you get farther away, you actually don't have this PI38.4, but microglia have this SX ceramide. Turns out there's no biochemical pathway even indirectly that converts these two lipids. So I'm standing here going, that's cool. I'll figure this out later. That's post-tenure needs problem. So I'm standing at the ADRC, and this is why you should give talks to like unique audiences. So I'm giving this talk, and this guy's in the front row, and he goes, "I know what's going on," and I go, "Please enlighten me. I would love to know what's going on here." And he goes, "There's a hypothesis actually that microglia scavenge lipids around them, and then once they scavenge a certain lipid, they become activated." And I was like, "Well, that makes a lot of sense, man. Why didn't you tell me this 6 months ago? Because I would have actually followed up." So what we're doing now is we're actually trying to study this with isotopes and flux experiments to see actually is microglia taking up lipids from the surrounding, and then where is this lipid coming from? But again, this all happened about a month ago because again we're fairly new as a lab, so I went a completely different direction than that. I said I'm a chemist. Oh yeah.
Microglia because of the matched histology information.
Yep. We stained with MXI or multifunctional fluorescence. We have a protein marker, and then we also have some mRNA. So it's all in the same tissue, so we know what's going on. So because I didn't know what that Alzheimer's researcher knew, I was like, well, I wonder if we can modulate this effect by diet, if we can actually control what fatty acids the animal has access to. So here on the left is a lipid that's kind of the same between both control and keto diet animals. We also have a Western diet, but that made me too sad because I like McDonald's, so we're going to ignore the data. I'm going to tell you it makes it way worse. We have our hexaceramide and we have our PI384. So remember, PI384 involved with activated microglia, probably scavenging anyway. On the top, you can see that's the image you saw before. There's lots of activated microglia, animals very ill. Turns out when we feed a keto diet, we have the same plaque burden, but there's a substantial reduction in PI384, and these animals are actually cognitively better than their control cohorts. Turns out you feed it burgers and milkshakes, it's much worse. So what I'm actually wondering is if the body's intentionally trying to undergo ketosis to stop or eliminate or reduce activated microglia, which again is involved in neuron pruning and actually makes you undergo cognitive decline. But we're also chemists, so I looked at the pathway of ketosis and I said, I can actually isolate or synthesize every intermediate that the body has in ketosis. And we actually found that DHB is the molecule responsible for reducing PI384. So if you feed animals just VHB, and that's a byproduct of ketosis, you get an even better result. So what we can do and what the benefits of this is one of the first projects we undertook, is we can start with a lipid that we know nothing about that other people said wasn't important and actually trace it all the way to something that might be therapeutically relevant, right? Even a molecule.
That's a quick question.
Yeah.
So I know there was some data about ketosis and improvements in schizophrenia as well. Is this the same mechanism or similar? These are microglia activation. It's really interesting.
I think it would probably be related because schizophrenia is hard, but because it's not entirely genetic, I would be unsurprised if there's some environmental factors that got microglia excited and then they started destroying neurons. But I think there is some benefit because this also affects astrocytes too. Microglia just are the worst as far as Alzheimer's is concerned. But my guess is ketosis is doing a similar thing. And we could study that too. There's fewer mouse models for schizophrenia, but we're starting to actually move into human patients. And so we have human data that's actually from Colorado, where individuals live their last 10 to 15 years in a very controlled environment where we actually know what diets they have. We're using MXIF to understand more what cells are doing in these environments. Again, with some things that are highly controlled or tissue that is less controlled, where we know that the individual is probably eating their McDonald's like meat. And so we're trying to understand, do these mechanisms correlate to humans? It seems like they do, and then there's more exciting things. So Alzheimer's disease, turns out what you need is important, and that's cool. That's something we're starting to figure out. But I also like to think about corollaries between diseases, because again, I'm a chemist. And so Alzheimer's is cool. We talked about the microenvironment around labs. You think about things like cancer, we have a very similar phenomenon. But instead of like, I don't know, 100 microns, say, we're talking about millimeters. So our areas in our neighborhoods have to be much larger. So this is actually a liver tumor. these are different molecules related to HCC, either the environment around HCC or the tumor itself. So instead of single cell layers, we looked at, we actually saw dysregulated metabolism 15 cell layers away from the tumor, even farther. So when you think about things in spatial biology, some of these events actually percolate quite far from the host site. But we can say play similar games where we're like, instead of an or a plaque, we can use the solid. We can say, here's the tumor, what's our microenvironment as we get farther away. We bucket this based on what our chemistry tells us. But what we can then do is we can say, okay, what is this actually doing in vivo? It's hard to do this in animals and see what we can do. But we can identify things like this PC38 was massively upregulated around the tumor interface. So what I wondered is, I wondered if the immune cells were actually producing it or the tumor cell was. And so we took a primary cell filter of the tumor, and then we fed it a lot of PC38-4. We have a lot of different abundances, but I'm showing you on clarity. And then this is growth of the cell filter. So at the beginning, it's actually massively increased growth of our tumors, our primary cell line. And that made some amount of sense. But then we actually started to see massive cell death, even though we fed it more. We didn't feed it more stuff. So I went back again to our big biochemical map. It turns out to actually metabolize PC38-4, you need cholesterol. So what we believe is going on, and what we're trying with some labeled experiments, is we believe immune cells are producing this lipid in abundance to deplete the growing edge of cholesterol. And in fact, when we knock out genes associated with the production of PC38-4, we actually see massive growth in tumors in animals. And then when we enhance the regulation of PC38-4, we actually see shrinkage of these tumors. So we believe immune cells are using this as a way to actually prevent tumor growth in situ. And so often the cool part about lipids and metabolites is their effects aren't direct. They're often indirect. But I think the body does some really neat chemistry to actually prevent things from continuing. And so there's often diseases that actually have no really hallmark spatial biology. So like in the case of HIV or SID, it's not like you form a plaque per se, or you have a giant massive tumor, but you have massive dysregulation, again, functional disease. So I work with a lot of undergrads, and we try not to have live HIV in our lab because they like to push the limits of how they do science. And so, said nicely, so instead we have macaques that are infected with SID, which is the monkey form of HIV. You can't get it. I don't stay up at night crying that my undergrad staff. But yeah. So we have uninfected kidneys, SID infected kidneys, and SID heart treated. So what I didn't appreciate after I learned talking to Tiana Williams, she's at Emory, is she said, you know, we should study HIV and SID. And I said, that's cool, but don't we have like, isn't that like kind of a solved problem? And she goes, no, turns out people on like ART treatment or antiretroviral treatment undergo kidney failure. And I was like, oh, that's a real problem. Like you don't want to do. And so what we wondered, and no one's actually really studied the impact of ART on kidney health with spatial biology. So here's our cool mass spectra and our images. I show this mostly to remind people I do mass spectrometry. Some of my colleagues don't believe me. They're like, you just show images. But we have multiplex immunofluorescence, which is a protein label. What we can do is acquire this on the same slide. What this allows us to do is segment our mass spectrometry data into things like distal tubules, collecting that, so on and so forth. Again, histological or functional units that have some medical or clinical utility. We can then start looking based on these functional units, based on or see their dysregulated metabolism. So this is a PC32.5, which is actually something I consider to be a housekeeping unit, something that just maintains cellular membranes. Many people don't think it's that useful. It turns out this is the hit actually that's SID infection. So in uninfected glomeruli, you get a pretty narrow distribution. Again, it's housekeeping. On SID infection, you actually get a much broader distribution of PC30. And then upon antiretroviral treatment, you actually still have this very, very broad distribution of PC32.5. And so I was puzzled about this. It actually is even wider than SID. And so when we actually go back to the spatial biology, it turns out that this is bimodal. I'm actually not tall enough to point at the slide. But we have half of the glomeruli have upregulated in SIV-like levels of PC32.5, and half are actually returned to normal. And when we look at these health based on a PAS stain, these glomeruli are inflamed. And so what we believe is the antiretroviral is actually damaging some of these glomeruli and preventing them from returning to normal metabolic states. Yep. She asked how long were they infected before starting art, and I believe I'd have to look. I think it's 3 months, but it's gonna be short. So the question is, do we think it's antiretroviral, or do we think it's SIV pockets, for lack of better, or like reservoir virus? And the answer is we're trying to figure that out. So what we're doing now is actually directly imaging the antiretrovirals. So this was actually a cocktail of three. So we're seeing where does that end up. We're also imaging the virus to see if there's any residual particles. And we're also exacerbating this with diet and specifically Western diet, which induces a lot of inflammation. We're seeing these human scores. So the answer is we don't actually quite know. But our goal is to kind of figure that out. Because if it is the the solution is actually similar, where we develop better antiretrovirals that either eliminate the viral burden in the kidney or design a drug that isn't so harsh. I could see it being both, actually. So we primarily use frozen, but we can also work with FFPE, just the question we get at a different. So lipids often are removed with FFPE, but in some of the metabolites can be, it depends on which ones we're looking at. Proteins are fair game and mRNA. And sometimes you can get lucky and get lipids. It just depends on the disease. But these are all great questions. And then we can do this again for proximal tubules, glomeruli, so on and so forth. Some get increased, some get decreased. So we're trying to understand what are these changes and can we revert them to normal? And this is actually improve health of the monkey. So another cool story in my lab involved the spinal cord. This was actually how Amanda emailed me finding this paper. And so, like all good collaborations, I met Aijun Wang in the park, day 3 of my PhD. And I tell you this story for the trainees in the room. I had this beautiful plan of what my science was going to be, and I've done absolutely nothing. Because partly I've met this man in a parking lot, and I randomly meet people at conferences. Deanna I met at a conference, and I was playing with her kid. The kid walked up to me, and I was like, yeah, you're 3, high five. And then we started SID. And so some of your best science is actually not pre-planned. So be open to experiences in science. But Aijun was talking to me because he's like, I just saw your talk. Oh, you're a new hire at UC Davis. That's cool. How do you like Davis? I'm like, it's great. I'm like, what do you do? He goes, I cure spina bifida. And I'll go, why are you talking to me? Go cure more spina bifida. I'm like, what are you talking about? And so he's like, no, for real. And he showed me this video of this child running around. And I'm like, for real, go cure, do the thing. I'm not important. I'm day 3. So anyway, he's like, jokes aside, spina bifida is a very debilitating disease that affects infants in utero. So you have your spinal column, and at some point you have a defect in your vertebrae, which causes your neurons in your spinal column to leak out. And then as babies do in utero, they kind of rock and roll, and they damage all of those neurons, and they're usually born paralyzed, right? And so Aijun was a surgeon, and in utero, he applied the stem cell patch to this vertebrae, and then this actually protects the neurons in vivo. They get reabsorbed by the vertebrae, which is really cool. And like 70 children now run around. It's really easy. They have spina bifida, not paralyzed. So of course, an analytical chemist, again, I go, so what's the variance of this? Like, as you're still talking to me, instead of doing more surgery like you should be doing, he goes, it's not perfect. I don't know why. It has nothing to do, seemingly, when we apply the deep cell patch, as long as it's early enough. And it doesn't have to do with things like sex or even help with the mother. And I go, that's cool. Why didn't you figure that out? He goes, that's a great question. no one really can image the spinal cord without decalcifying it, adding nitric acid. I was like, well, that's cool. I got nothing else going on the rest of the week. I'll take one of my students that joined on Hopes and Dreams and said, why don't we try dissection? So a couple tape methods later, and some migraines, this is her first image. This is completely unfixed, undecalcified, non-touched spinal column from an adult mouse. So in red is the muscle, yellow and blue's white and gray matter, blue is the fluid around your spinal column. Green is blood vessel. You can actually trace this through. Pink is bone marrow, and black is actually the bone. So there's some cracks, but broadly speaking, you can get this native chemistry. And again, the power of mass spec, we detected a bunch of stuff. We can then histologically stain this after shooting lasers at it. People ask, is it destructive? I mean, honestly, the razor blade is way more destructive than my laser. So we can get some histological stuff and build profiles. But what's even cooler is we can start actually feeding animals retinoic acid, which sometimes randomly, sporadically results in spina bifida. Again, we don't understand this at all. We're still trying to understand this. We then section along the defect, and we're starting to understand what is chemistry changing, and can we use this to design a better gene therapy. So turns out when you feed animals retinoic acid, or you don't, you get healthy spinal cord development. This is a different angle than you saw, but this is what that looks like. Most animals, when you feed them retinoic acid, their pups have inflammation, they're antiseptic, and so we have a high abundance of this. And then I think 1 in 10 or so have this defect formed. And what we see is actually you get massive alteration of metabolites that are associated with food bonds or polyunsaturated fatty acids. And so what we're doing now is understanding if we actually supplement the diet with our gene therapy, can we actually treat more cases of spina bifida? So again, met this guy in a parking lot. Oh, that might be a good place to go. We've tried a little bit of that, not as much. The reason we went with retinoic acid is it had it in lab already, but inflammation is also not bad, because it would be good to see even if you had with 100 mice with anything develop spina bifida. We still don't understand what causes it. It turns out, like, we tell mothers to take a lot of folic acid. Turns out if you take too much of that, it also causes spina bifida. And so understanding these things is really important. And so we're trying to understand, like, what is the magic number? How does that vary? What are some of the endogenous factors that can actually do this? And then try it on multiple things, I think is important. And multiple models. We're also doing this in sheep and dog, and then human. So we have some, this is all mouse tissue, but we're doing this across species. Dogs apparently also form spina bifida, which I did not realize. So there's bulldogs that we are treating as well. And so I've mostly shown you organs that are removed from the animal species. But if anyone's ever met someone with Alzheimer's disease or cancer, it's a whole body disease, right? And so we focus, I think naively, on the organ that's mostly impacted. But I think there's a lot of information we can gain from some of these peripheral organs. And so I'm moving a lot of this biology to actually do whole organisms. So here's a whole mouse, and I'll show you an actual one next. So if you don't like it, just cover your eyes. Here in red is a lipid that actually stains the blood-brain barrier. So we're visualizing astrocytes here. Neurons. You can see the muzzle. Yellow is your spinal column, so on and so forth. So whole animal, spatial lipids. Here's the animal. Close your eyes if you want to, but you're like clinicians. Or clinician adjacent. So we have an adult mouse, and we have polystyrene. So I didn't want this experiment to work. Karina did, and it's one of those times as a PI where I'm like, good on you, but also this is terrible. So Karina is from Mexico, and she cared a lot about environmental exposure, including water. She goes, I want to look at exposure of plastic. And I said, excellent. We can try that. No one succeeded so far, but you know it is important. And she goes, I want to feed mice plastic or food out of a styrofoam container like a picnic. I said, cool. There is no way that's enough plastic to actually detect it. So I was wrong. Fully admit that I was wrong here. But we detect polystyrene from food kept in a styrofoam container. So this paper was recently published. We did convert from the styrofoam container strategy to Gavage plastic, so we knew how much we were going to be in. It turned out that the amount of plastic in the food was difficult to determine, but it was enough we could detect. So there's a couple key points here. On the top is an animal that was not exposed to polystyrene, and on the bottom is an animal that was. So as you can notice, yellow is high abundance. Our control has an amount of polystyrene. We spent a year and a half trying to find a control that did not have polystyrene. What we've done so far is over the Bay Bridge, there's animals kept in like a containment facility that are controls for smog and stuff, and they also have plastic. In fact, if you Google, you can find plastic in sediment that like was made a million years ago that also has plastic in it that has not been unearthed in like a million to 2 million years. So no matter what you're looking at, your controls all have plastic in them, and that should scare you. It scares me deeply. So this is actually the original experiment where that was this animal was fed food out of the styrofoam container and the top of the top. The next slide in the data is based on a more controlled experiment. And so the weird part about plastic is that it's a polymer. And what we've assumed as chemists for a long time is that the polymer just gets like excreted in some way. And the body doesn't interact with it. So when we actually looked at our animal after exposure, we found a lot of peaks we had no idea what they were. None. They had weird mass defects, which is a mass spectrometry term that tells you something about class. And so I was like, this is probably worse than I thought. Not only did I not think this was going to work, but also I think the body's actually metabolizing plastic, and that's also wrong. So what we did is we took a lot of standards, and there's no good standards in plastic. And then we used ion ability to actually characterize gas-phase characteristics, so we can find molecules and then say they act like polystyrene, but they maybe got something that's weird. We then do orthogonal validation that in fact they're polystyrene. Cool. So everything in red is a molecule that it has a polystyrene backbone that the body has done something to. So that's great. So we go, excellent. So again, not biologists here. I go, we're orally exposing these animals to polystyrene. It's going to be in the stomach. And so again, what most people have done is excise the stomach with the polystyrene plastic. So what we figure out is we look at a dimer of polystyrene, so a small component of polystyrene, which happens to get mechanically broken down in the ocean all of the time, which is so wonderful. You then see a high abundance in the stomach and the intestines. We go, yeah, it's orally exposed. Cool. And then, did you smell the other yellow? So it turns out this speck here is actually where it's caused leaky gut. So the polystyrene causes leaky gut, leaks out of the gut, and actually ends up in the bloodstream. And once it ends up in the bloodstream, it gets pumped to the heart, which is that yellow right here. Based on how we section this, the heart's kind of in that plane. And then once it ends up to the heart, what we believe is happening, it's getting pumped to the lungs, the adipose, and we've seen it in the brain. And the cool part is it's actually modified. It's a monomer of polystyrene that has a couple OHs, so it enables it to cross the blood-brain barrier. So the power of mass spectrometry is you don't actually need to know what you're looking at to actually see it. And without looking at a whole animal, you'd actually miss the fact that it leaked out of the gut, right? You would have saw polystyrene in the intestines and said, cool, stomach cool, and called it a day. Instead, we looked at the entire animal. We actually did this in 3D, so we checked all organs. And it turns out, this is where it's actually ending up and how it's probably getting there. We're starting more experiments to see that as well as it's actually clearing answers it's not clearing. And then, fortunately for us, it's facetious. When we measure the lipids in things like the heart and the stomach, you actually get massive inflammation.
The modification that when you look at the brain and the addition of some other molecules, that's specifically for the brain or that it moves from the gut.
There's more modifications that occur as it goes down. Specifically, what we found is there's no raw. Oh, sorry, I should reply. He asked about the modifications that occur throughout the organism. So the reason I point out the brain is we didn't see polystyrene, just polystyrene in the brain, so we thought it wasn't getting there. But when we actually saw things that were not in our control that were in our exposed. So when we actually did our gas-phase characterization and the fragmented, we found out they had a polystyrene base. structure, which is not big. We don't create that. But we found OHs and stuff, so we think that P450 is actually active. And that's causing it to be blurred up to cross. But this is very preliminary. This is something that we did not expect, and we're still trying to chase it.
That's terrifying if the CIDMI system can modify plastics to cross the.
Yeah, it's not great. It's not good. I will say that's a hypothesis. We're not validating that. But it's got OHs, and that's one of the enzymes. So when we look at actually lipid remodeling, we get an abundance of lipids that are associated with inflammation. And so what we believe is upon exposure to polystyrene, you get inflammation and even some oxidative stress on the organ. But there is good news. We found organs that don't actually get polystyrene through this exposure mechanism, and they actually have completely normal metabolisms. And so what we're proposing now is that this is true, and you have to actually directly interact with polystyrene to get inflammation and oxidative stress. What if we designed a plastic that doesn't end up where it's not supposed to be, right? It's either resistant to mechanical degradation or enzymatic bacteria. Then you just localize it to whatever you're actually where it's supposed to be, and then it will get extruded. So what we're doing is building a giant database of plastics and seeing where they end up and trying to trace and figure out biochemically how they're being altered. Again, this was just published last week, so this is new. So we have not validated this yet. Yeah. We're figuring that out now. So in some cases, we know or we believe that they're not there. So in the case of the spinal cord, I don't think they're making it there. How or why, not sure. So what our running hypothesis is that there's some chemical component of plastic that enables it to bypass some barriers or be altered. And so by building a library, we can then find features that are enabling, like if these three plastics end up in the brain, clearly we don't want to use those plastics, or they're shared chemistry. And so we're still early understanding this. We just know if we don't detect plastic, they actually don't have altered metabolism. This is actually true of sorry. So this will probably be the last project I'll talk about, which is cool. So we've talked about giant or whole organisms, but things actually happen on the teeny tiny scale. So there's these molecules, these features called extracellular vesicles, and these are small vesicles that actually bud from cells, from their membranes. They're used to do long-term signaling, trafficking. In fact, cancer metastasizes, we believe, through extracellular vesicles. So I did some math. I said we should actually be able to detect stuff from single EVs. Sure, they're 30 nanometers, but you can also detect 30 copies of things, and that's about what a 30 nanometer molecule is. And so here's actually the spectra from a single extracellular vesicle. So our noise is about here. So this is very low signal to noise, and I appreciate that. But there's some lipids that are actually high, threefold where signal to noise would be. They actually map back to the amount of molecules you would expect from something that's 30 nanometers. And before anyone says anything, we validated that this is an EV with electron microscopy. So I actually know that this is EV. We've done a lot of synthetic EVs and validated the system. So this is truly a single EV. We've analyzed about 1 million single EVs now. But what we're doing with this is we're trying to see if we can do blood-based diagnostics. So I hinted that cancer seems to metastasize. And how that metastasis occurs is probably by EVs budding from one of the tumor cells going in the bloodstream and ending up somewhere else. So what I thought with Randy Carney is I was like, well, we should be able to see that it's budding from the membrane. They should share a lot of chemistry. Tumors have very unique lipid profiles because they grow and proliferate so abundantly, so quickly. Certain lipids allow you to do that. So on the top is ovarian cancer tissue, actually mine. Long story short, we started this and I was collecting a bunch of tissue. I ended up with a 15-pound tumor, and then I was already collecting tissue and got my own tissue. So I show this on the top because what I get for feedback from the NIH is that we've solved ovarian cancer, and the answer is no. Partly because it's a women problem, and then partly because it's actually challenging to diagnose ovarian cancer. So I got to be 15 pounds. So before anyone says that ovarian cancer is solved, it's not. So in this tumor image, we have a lipid in red, which is actually the necrotic core of this tumor. There's multiple tumors in this ovary. In blue is the growing margin, and then in green is the healthy ovary. There's not much, right? All of these cells are spewing EVs. So what we do is we analyze about 1 million EVs in the blood without any treatment. And we find EVs that actually co-localize with some of these lipid features. What this is allowing me to do in our expanded patient cohort is actually do staging and then organ of origin, because again, the lipids are unique. So what our idea is that if we can actually just take blood, we might be able to do early diagnosis. I'm not saying this is the end all be all, but it might give patients what they need to go advocate for themselves to get additional testing, especially in difficult to detect ovaries or difficult to detect diseases. And then finally, I hinted that I want to do spatial multi-omics on one instrument. I usually make a joke that you usually need a startup of $1 billion to buy all the instrumentation you need to span the central lab of biology. But I believe mass spectrometry is cool, and we might be able to hijack some traditional molecular biology to actually do and span the central lab of biology. So Kyle was one of my first students who worked for 10x Genomics. He was telling me all about how cool busy it was. And I said, well, what if you did mass spectrometry? And again, he bought hopes and dreams and then joined my lab instead of all of the transcriptomics people data. And so I was like, we need to do it better. We can't just keep spending like $10,000 and hoping the data's good. So him and I sat, we're like, the original way of studying RNA expression is through pitch. So that's like terrible because of spectrum crowding, blah, 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 which mass spectrometry conveniently does not have. So instead of fluorophore, we do fish assay, but instead of fluorophore, we actually add something that's mass spectrometry reactive, allowing us to actually detect indirectly where these mRNA are. This is what the data looks. So in red is GFAP, which states astrocytes, and NFL is a gene that correlates to neurons. On the top, Kyle's like, look, I did it. $500. There's per gene. You can do this for 100 tissues. And I said, Kyle, this is great. This is cheaper, blah, blah, blah. We're functioning on hopes and dreams. Got to keep that mission alive. Hopes and dreams, no resources. So I was like, you're a chemist, make it cheaper. So we redesigned the entire thing. So now we're at $15 a probe for 100 tissues. Complex, hundreds of mRNA. And so here is FOXP2, so we can see its expression in the cerebellum, within a single cell layer in the hippocampus, and multilateral where it's supposed to be for very large pieces of tissue. But again, we're doing it all in a mass spectrometer. So we can do it to detect mRNA at the same time as lipids and metabolites. So on the top here, there's two lipids, PI 38.5, PI 36.4, and red, which is GPR, and which is dendrocyte. So I'm just showing you can do them at the same time, same instrument, sagittal section. You can do it on the whole animal if you chose to. Again, $50. So here is an oligodendrocyte. Here's mRNA profile, and here's the lipid sequence. So I hope with that showed you that we can span from extracellular vesicles all the way to the entire animal, and that there's actual cool stuff you can figure out at each of these different steps. And that I hope I've convinced you that maybe we can actually do span something about biology on one instrument. So far, we've done mRNA, proteins, and metabolites, and we're starting with some DNA. So chromosome tightening and stuff. So with that, I once again want to thank my students for functioning on hopes and dreams. Some of our funding sources, I scrape a lot of money from industry because they're like, wait, you could do that? And I was like, yeah, we can. And then I'd also love to thank our clinician collaborators because they tolerate my nonsense. So with that, thank you for listening. Here's my email in case you have questions that you're too afraid to ask, or if you have ideas. So thank you for your attention and time. I appreciate the very informative talk.